All right, so this lesson is on human immunity. Okay, we're looking at chapter 31 of the textbook. And it's a nice thing to throw in with the cell cycle because we've been talking about all these cells and we've been looking at them in, really close focus kind of view, right, as far as their molecular machines and all that. But we haven't really talked about something that cells do for us in the body. So we're going to look at human immunity as an example of the important roles that cells have when they interact with each other. So let's get this here in. So some questions we want to make sure that we can answer. We want to know what causes disease, all right, in human beings and organisms, whatever. We want to know the systems and the structures that form the immune system in human beings. We want to know how the immune system responds to pathogens. We want to look at issues that occur with our immune system. We want to see how we can support our immune system. And we want to look at what can lead to a weakened immune system and the outcome of that. So we're really going to focus in on the immune system here. Now, germ theory is the theory that microscopic particles cause diseases. It says that, look, if you get a certain bacteria in your intestines, you get diarrhea. Or if you get a bacteria in your brain, then, you may end up with encephalitis, right? It says that these bacteria or germs, other microorganisms, cause disease. It was first proposed by Louis Pasteur, right, the same guy who came up with pasteurizing milk. It led to some really fast advances in understanding disease. Once people knew what to look for, they could find what caused the disease, and then they could try to kill it. And a guy named Robert Koch, Koch's postulate supported the idea. And Koch actually had some really a really simple way to determine if something caused a disease. So let's say I found, this will be our little bacteria, right? It's got a nucleus, it's got little, right? Let's say I take the blood of a sick person and I find this. So we have a test tube, right? There's our test tube. It's got, we have a blood sample. And it might be a spit sample or something. And we find this bacteria. And we know, hey, that bacteria is not supposed to be in people. So what he did is he put this into a needle, right, into a syringe. This is like the worst syringe ever. There we go. And you grab it, push it down in there. It's got those little wings out on the side. There's the needle right there. So you put the bacteria in there, and you're not going to probably like this part. He put it in a rabbit. Right, there's a rabbit. And he waited to see if the rabbit got sick. And if the rabbit got sick, then he took another blood sample, right, from the rabbit. So he took rabbit blood and he put it underneath the microscope. And he asked a simple question. Is that same bacteria there? And if it was, he said, okay, well, we found this bacteria in the sick person. We found that same bacteria in the sick rabbit. That bacteria must cause the disease. He called those disease-causing agents, he called them pathogens, all right? So that's the word we use. We don't usually, we can say germs, but pathogen is the more correct terminology. And there are a lot of different advances that took place after germ theory. But it's kind of interesting to look back before germ theory, what people like to do. So sometimes we'll find skulls like in South America, and they'll have a hole drilled in the top of them, like right here, that hole. And that's because people are like, oh, you have a headache or you have a tumor or something. Like they didn't know about that. They were like, oh, you have a spirit in your skull, so we're going to let it out. And then drill a hole in somebody's head. Later on, the Greeks thought about the humors, like all these different fluids in your body. And if they got out of balance, you could get sick. They didn't really start to study anatomy until the 1400s. This is just a picture from the 1500s here. And, people were like, it was kind of taboo to cut up dead bodies, right? It messed with people's religious ideas, so they didn't really do it. Germ theory, though, happens right here in 1857. And you can see before 1857, right, all of human history, we're not really doing a great job treating disease. After 1857, when we go, oh, it's these microorganisms that are causing disease, now stuff starts to happen, right? So Joseph Lister comes up with the idea that if you clean your surgical tools, patients won't get infections, right? So before you cut somebody open, you should probably wash your scalpel. Robert Koch came up with those 4 conditions that prove a pathogen causes disease. There were, 1900s started to see the application of antiseptic. Alexander Fleming discovered penicillin in 1928, which was one of the first antibiotics. We, graduated from looking at bacteria to also looking at viruses. We came up with one of our first vaccines that got rid of polio on a massive scale, one of the first massive scale vaccines. And as time progresses, right, all of these wonderful advancements started with germ theory. So there are a couple of different kinds of pathogens we want to be aware of. First, we have bacteria. They're single-celled organisms. They usually cause illness by either destroying cells or by releasing some kind of toxic chemical in the body. There are viruses, which we've talked a little bit about. They are genetic material with a protein coat around it called a capsid. They're going to force host cells to make more viruses, make more copies of themselves. But the problem is they're really small, and so they're very hard to find. Okay, this would be an example of a bacteria, and there's an example of a virus. Now, this and this, they look like they're the same size, right? So there's our virus, and here's our bacteria. These are pictures taken with very different microscopes, right? So this one here, this would definitely be either maybe a scanning electron or a transmission electron microscope. I'm not sure which. Now, other things that can cause problems include fungi, protozoans, and parasites. So fungi are like mushrooms, right? Or molds. Those are examples of funguses. Fungi can cause disease in humans. Protozoans are single-celled organisms, usually eukaryotes, that are going to steal nutrients from their host cells or use the host cell to complete the life cycle. So they may need a host cell in order to reproduce or some other aspect of their life cycle. Parasites are multicellular organisms, and they're going to grow and feed on the host. They might even kill the host. Okay, so this one is a trypanosome. I think I spelled that right. This big guy right here causes malaria. All right, very, very, very dangerous disease. But that trypanosome right there would be a protozoan, and it gets inside a red blood cell. It eats that red blood cell, kind of hollows it out, hides from the immune system, makes a bunch of little protozoans, and then they bust out of red blood cells. You get really tired and sick, and then they all burrow into a new red blood cell, and you start to feel better. But then all at once they bust out again, and you might die the second or third time that happens. This is a parasitic worm. One of the things you can see about worms are segmented. Just like an earthworm is segmented, right? You can see all those little segments there. So, things like hookworms and heartworms and tapeworms, those are all parasites. They're multicellular organisms that live in the body and steal nutrients. There are a lot of different pathogens. This is a pretty important list, so look over this, maybe write a couple of these down.